Uh, I, I appreciate your patience for those of you who know this material very well. Maybe for a few of you, it'll be new. And having just heard the previous talk, I maybe should be emphasizing slightly different things. So we'll see how that goes. And the other thing that I'll need to ask your patience with is that I have COVID and I'm not feeling the best. So I appreciate you understanding that my voice is a little bit hoarse and I'm also a little bit out of it. So I appreciate that. So I want to talk a little bit about connecting the kinds of network modeling that this course has been focusing on for the past week with the kinds of multiscale world that I live in. And I should say that Herbert and I have worked together for many, many years. So this is a long standing collaboration. And in the past few years, we've become more and more focused trying to understand what it takes to do improvements in actually delivering of biomedical understanding and treatment. I think we're still a bit far from that, but at the back of our minds, when we do this kind of modeling, ultimately we want to do both scientific exploration and discovery, but eventually we'd like to be able to deploy these kinds of tools in practical biomedical applications. Here, the context is always going to be multicellular tissue level organization. Classically, problems in embryonic development. How do you organize an animal starting with a fertilized egg? Homeostasis, how do you maintain an animal once you've built it, or to be more precise, once it's built itself? And then what are the failure modes of that organization? Developmental diseases like cancer, where the normal homeostatic mechanisms get deployed in pathological ways. Infectious diseases, where you have something coming from outside perturbing the system, and the system needs to respond to protect the system. Potentially, how do you design therapies and improve them? And how do you build totally new things if you're doing bioengineering, like tissue engineering? And then, as we heard mentioned, that we do some toxicology, how do molecular perturbations affect systems level outcomes? In all of these cases, we have to think about interactions at multiple scales, interactions of physical, chemical, and biological signaling processes. Ideally, we want to be able to predict things, and if we can predict things, we hope we can design control strategies for them. We're never going to be able to model all of the scales in an organism simultaneously. Trying to go from a single atom up to a cell is already out of reach. Every cell in the tissue is out of reach. And so we have to pick subsets of scales that we're going to work with, depending on the problems that we're going to address. What we call virtual tissues typically take a 3 or 4 scale approach. We start out with network models of gene regulatory networks, metabolic networks, signaling networks inside of cells. And we have to remember always that a network model is already a very harsh abstraction because it's ignoring the spatial organization that's going on inside of a cell. And that may mean that the predictions of a network model are not always accurate. Another thing that's difficult is that we have to connect the outputs of a network model, which typically are things like chemical concentration. We have to connect those outputs to the behaviors of cells. And that's not necessarily something that we know how to do in an organized way. Once we have cells as agents, because cells are, in fact, in most cases in biology, the governing agents in the system, we need to be able to understand how those agents interact in a spatial context. And we have to remember that they create the environment that they respond to. And so there's a lot of coupled tight feedback within and between scales. And we'll come back to this example in a minute. The network modeling happens, of course, at the smallest scales within the cell. We also tend to use network models to do things like PBPK pharmacokinetics and transport at the whole body level. And so we typically have network models of the kind that you do in this week at the small scales and at the large scales, and then we have multicellular models in the middle. Multicellular models are mechanistic, they're dynamic. These are agent-based models. And so one difference from classic Antimony or tellurium is that we have to run many replicas of the same network because every cell will own its own copy of the network. We're focusing on emergent spatiotemporal phenomena resulting from interactions between agents, in this case cells typically. These are mechanistic models, they're additive, and typically our outputs are spatial time series. And one of the questions we always run into is how do we quantify our results? How do we know if a set of results match experiment or not, given that the world is stochastic? When we're going to represent cells, there are many different ways of representing cells. You could represent them as cellular automata. You could represent them as point particles that I know you had Paul Macklin and his center model method of Physicell presented earlier today. You can use the techniques of solids modeling, finite element methods, and have very detailed models of the boundaries of cells and their mechanics. The one that we use, GGH CPM, is a stochastic lattice-based model, and it has the great advantage that effectively you can think of taking a digital picture of a group of cells and you simply label each cell. So all of the pixels that belong to cell number 1 have a number 1 associated with them. All the pixels associated with cell number 2 have a number 2 associated with them, and so on. And so there's a very direct mapping from an image of a tissue to its computational representation. The kinds of modeling we do have a lot of basic concepts. Again, I'm sure Paul covered the concept of a chemical field and the diffusion, decay, secretion, and absorption of a chemical field. Our cells have intrinsic properties like volume, surface area. They have behaviors like cell motility, growth. They have the ability to die and to divide. They can also interact with each other by adhesion, force generation, and they can interact with things like chemical fields by secreting or absorbing chemicals, differentiating in response to chemicals, or moving in response to chemicals. There's also a solids concept of a link, which can represent things like junctional adhesion or philopodia. And then the network dynamics that we've been talking about in this class for the past week. So these are the basic groups of object classes that we're going to be modeling. We developed the Computational 3D simulation environment. And the idea of Computational is to try to make modeling relatively easy. These days, we probably wouldn't do it this way. And actually, I think Paul, if he showed you some of his GUIs, has actually done some beautiful things with Physicell. In the days when we started Computational, a lot of the technologies that are available today weren't available. And so Computational currently primarily exists in the form of a customized editor called Twedit, which has, like using, say, Spider to do Antimony models, has a pretty good knowledge of what the languages are. And then a player, which is a tool for visualizing, controlling, and exploring, executing simulations. It is downloadable cross-platform for Windows, Mac, or Linux. Based on what we were just hearing a minute ago and what I'll show you quickly today, it also exists as a cluster and cloud executable service. There's an AWS version, and we run a lot on something called nanoHUB, which I'm going to show you in a minute. And I'd encourage people during the talk go to https://nanohub.org/tools/cc3d base 4x. And if you can do that, let me grab that link for you. One of the things that we like about nanoHUB is that it's free, it's open to anybody, and you can create an account. You can create an account in a few minutes if you like.
James.
Okay, thank you. Oh, Hayden, you're there. Thank you. Okay. Computational is a flexible software tool. It works. It's not used as much because multicellular modeling isn't as big a world as network modeling. But you can see that there are quite a few publications using it in the past couple of years. And some of these are from our group, which is maybe not surprising. We're much prouder of the ones that were done by people we've never heard of, because that means that people are really using the code effectively. I want to give you a couple of quick examples, and then we're going to go explore the tool a little bit. There's some classic developmental biology examples. I mentioned, you saw this one already. This is replicating the formation of the vertebra in a chicken embryo, the pre-patterns of the vertebra, actually, the segmentation patterns called somites. What's nice about this from the point of view of a network problem is that you have quite elaborate signaling and gene regulatory networks inside of each cell in the presemitic mesoderm. These lead to complex oscillatory behaviors. Those network oscillations are coupled between adjacent cells, leading to synchronization of the oscillators between adjacent cells. There also are both anterior-posterior and posterior-anterior gradients of a variety of morphogens that lead to modulation of the oscillators and also lead to the cells in the tissue reading the phase of the oscillator when certain chemicals cross thresholds and differentiating in response to those thresholds. And so what you see here in the simulation, which you've already seen played before, on the right, we see the oscillation of a particular intracellular species, a lunatic fringe. In the middle, we see the extracellular concentration of fibroblast growth factor 8, FGF8, which is one of the determinants of cell differentiation. And on the right, we see the differentiation states of the cells. They're going to change their mechanical properties. It's going to lead to the breakup of this continuous tissue into ball-like somites. One of the things that we found experimentally, along with our collaborators who did some beautiful experiments over many, many years, University College London, Claudio Stern and his group, led experiments which showed that you could, despite the fact that in vivo, these oscillations are definitely there, these signals that induce differentiation are definitely there, they showed that you could still get somite formation even if you turned off the oscillations and didn't have the induction signals. And that led to some work trying to understand the biomechanics of segmentation by Giulio Belmonte, who's now on the faculty at North Carolina State University. And he was able then to build a model of segmentation based on lateral induction of organization. In this case, there's essentially no subcellular model present. This is purely a contact signaling mechanism. So it's rather different from the kinds of models we've been working with here in this week. But it shows the self-organization going on here. And then there was some additional work trying to understand this in the context of the tissue, looking here at another kind of mechanical instability due to apical constriction. As I mentioned, to assume that cells don't have spatial structure is a pretty harsh one. This is an example again where there's no explicit network model running. All of the organization is happening because of spatial reorganization and anisotropic force generation. Another kind of example of what you can do with this kind of simulation is work. Over the years, we've done a lot on angiogenesis, blood vessel growth, in normal and perturbed situations. In this case, we're modeling the invasion of the retina by blood vessels that normally live behind the retina. In this case, the presence of a small gap in the basement membrane that normally keeps the blood vessels out of the retina, and a defect in cell adhesion leads to invasion of the retina, proliferation of the vasculature within the retina, retinal detachment, and eventually retinal breakup and blindness. This is old work, but I think aesthetically quite attractive, and something we were quite proud of at the time was that the time scales for the simulations actually matched what was observed experimentally or clinically in this case. And here you're going to see the final failure of the retinal organization, the loss of the photoreceptors. One of the things that you can do with this kind of model is you can predict the risk of particular patterns of disease happening. And so this was a study where we looked at different kinds of adhesion defects and their possible correlation with particular phenotypes of this disease. It winds up that the blood vessel invasion can happen in different patterns in different individuals, and those patterns of invasion are associated with particular adhesion defects, and that potentially leads to improved approaches to therapy for this disease. And this is work I would have loved to continue actually, but our prediction that adhesion defects were fundamental was not popular when we made it back in 2012, but it was validated experimentally actually a few years later. Another kind of thing that we've worked on over the years is autosomal dominant polycystic kidney disease. This is a proliferative disease, a little bit like cancer, but it never becomes cancerous. In this case, the cells in your kidneys continue to proliferate, and they form cysts, and eventually you die when your kidneys get to the size of watermelons. At the moment, there really isn't a good drug therapy. Kidney transplants are about the only thing you can do. In this case, it was understood that there were two gene mutations that were causal to the disease, but how those mutations led to changes of cell behavior were not understood. Our collaborator, Bob Bacalhau of Indiana University School of Medicine, had done some beautiful in vitro work suggesting that perhaps, again, adhesion defects were central to what was going on. Again, Giulio Belmonte did some nice simulations of what would happen if you took a kidney tubule, you introduced a cell with an adhesion defect, and he was able to show that you get proliferation and cyst formation. The morphology and dynamics of those cysts depended on the particular signaling mechanisms and adhesion defects that were present, and we're able to rule in and out certain mechanisms that were possible in the disease. And that was then combined with more classic bioinformatics analysis to suggest some drug targets that we're evaluating now in animal models. The past 2 years, like many people, we've spent a lot of time. Paul probably showed you simulations of tissues as well and immune response. This is a CompuCell simulation of a patch of lung tissue, starting with an initial single infected cell in the middle. That infected cell will secrete virus. The virus will spread, infect other cells, which will then go on to produce more virus. The cells will send out cytokine signals that will call in immune cells. Those immune cells will kill infected cells. They'll secrete their own cytokines to pull in additional immune cells. they'll also secrete oxidative agents that will kill uninfected cells as well. And this is a model that involves both detailed network models, because the viral life cycle is modeled using antimony in this case. The viral replication life cycle is modeled using antimony. And then you have multicellular layer, chemical field layer, and then also the activity of the lymph nodes producing the immune cells is also modeled using a network model, not running inside individual cells, but running in the whole simulation independently. This one is available for running online if you like, and I'll come back to running things online in a minute or two. You can use this kind of model to study things like the possibility of using antivirals. The first antiviral that was released, of course, was remdesivir, which was an RNA synthesis blocker, or actually an RNA elongation blocker. And you can simulate what happens with different doses at different times of initial delivery, because one of the things we know about antivirals is they really only work if you give them very early during the course of infection. You're able to understand the effectiveness of treatment. Again, these are stochastic models, so you'll see lots of different lines that represent here viral load as a function of time for different replicas. In this case, red means a failed treatment, green means successful treatment. The white in the middle represents regions in which sometimes some individual replicas are successful and some are not. You can put this in more detailed biochemical context, study the effect of dose interval, for example, on treatment efficacy. This is a paper by a grad student here, Giuliano, trying to use this kind of methodology to design more effective dosing schedules for these kinds of drugs. I mentioned that we have models of the viral replication, relatively simple, but nevertheless significant models of viral replication that run inside of each cell. In this case, this was work done by Shoemaker and Weaver at Pitt in conjunction with Josh Aponte Serrano, who's now a postdoc at NIH, trying to understand antiviral resistance induced by interferons. In this case, they have a subcellular network model of signaling and gene regulation inside the cells. There's a model of viral replication, and then an extracellular model of viral spread. You can put that into a spatial context then and model plaque assays, which are an in vitro assay for evaluating viral infectivity and control. And this is a paper that uses that. So these are all examples of mixing network models with multicellular models. Perhaps the most elaborate effort of that kind that we've done is this paper of TJ Segoe in our group, taking an ODE model of influenza, a very detailed, validated model of influenza, putting it in a spatial context. So asking the question, how do I translate a network model which has a representation of population, for example, of immune cells of a given type, and give a explicit spatial representation of those immune cells. That first model that I showed was quite simplified. It only had one immune cell type. It only had one chemical signal. In this case, they're explicit models of neutrophils, CD4 plus T cells, B cells, antigen-presenting cells, and 8 explicit immune signals, TNF, IL-10 and 12, type 1 interferon, and type 2 interferon. And so this allows you to then begin to explore the the significance of spatial organization on these kinds of models. We use this network model as a basis because it had been matched, parameter fitted very carefully to experiment. And so the parameters then we worked very hard to make sure that the parameters that were derived from the network model could be imported directly into the multicellular model. So now I'm going to show you, now that we've given a few examples of what you can do with this, I'm going to show you the code, and I'm going to show the code in the context of nanoHUB. And again, if you're able to get into a nanoHUB account, you should be able to follow along. If not, it won't take that long. You can try it on your own. nanoHUB, again, is a way of releasing simulations to be run publicly. It's not designed for doing research simulations in the way we were just hearing about in the previous talk. It's more for educational and exploratory simulations, although you certainly can do research on it. It's also a great way to publish simulations. And I should say that while I'm going to be talking primarily about CompuCell simulations here, I should say that there is a nanoHUB tellurium that's available online, which you can upload and download any tellurium models from, run them dynamically online. And in fact, I taught a class this week, a one-day class on Tuesday on network modeling. Well, Herbert, you were teaching network modeling here. I was doing a one-day network modeling class virtually for Purdue, and we used this online tellurium implementation inside of the Jupyter Notebook, and that's quite convenient. I'm hoping that my screen share is working and you're seeing a split screen at this point.
Yeah, we see that.
Okay, good. One of the things that you can do, and again, if you build network models in Jupyter Notebooks and you want to release them publicly, you can do that quite effectively. And if, for example, here, within the nanoHUB environment, I can search, and some of these PhysiCell models, these are ones that Paul Macklin would have shown you today using his code. But these are things like modeling a COVID pandemic with BSVIR-ID model. That's actually a released tellurium model. And so any tellurium model that you develop, you could explore and release on nanoHUB. When you put things up on nanoHUB, you get a DOI. The good side of that is that it counts as a publication and it's available, it's persistent, at least as long as nanoHUB exists, which has been quite a while. I assume it will continue to exist for a while. It's an NSF-funded project out of Purdue and now UCSD. And you can also get statistical information on usage, among other things. So if you go to the CompuCell page, it's nanohub.org tools CC3D base 4X. And I'm going to go there. Let's see, here it is. I have to find it now. Which one? I had it open earlier. That's the tellurium. There it is. And one of the things that's rather nice about this, you'll notice it's got a DOI here, so it's citable. And if you want to know usage, I can click here and it'll tell me how many total people, unique users have used the software, how many simulation executions there have been, and it'll give me a rough geographical map, assuming people aren't spoofing their URLs of where the users have been. So that documentation can be quite helpful if you're trying to track usage of your software tools. So it's something to think about as a deployment release mechanism for your methodologies. Anything you do with Antimony tellurium, you can release very, very easily on nanoHUB. Maybe it takes an hour to upload it to nanoHUB. It doesn't have quite the nice features that we were hearing about earlier. It's a little clunkier, but you can do a lot with it. So I'm hoping that people are able to log into nanoHUB. If you're at a US institution, you go to an affiliated institution, almost every institution in the US has it. Put your regular credentials in, you should be able to get into nanoHUB. Once you're there, you can use the dashboard, which you've already seen, to navigate. If you're there, you can search CompuCell. If you use the link that I gave you, you don't have to search it. Once you're inside of it, you can favorite CompuCell tool or the Tellurium tool if you like. Hit the icon here to execute it, or here on the home page, I can hit Launch Tool. It takes a minute to launch. Here is an executing version of CompuCell. It takes a little bit to get started, but this is the window of the CompuCell tool, and it's now pulled up the CompuCell player, which is the simulation execution environment. You can also, of course, download CompuCell for your computer if you'd like. And so what I want to walk you through is how to build a CompuCell simulation, and then I hope to take a few minutes to look at the code together. And we're going to start by launching Twedit, the model editor. I do that by either hitting the piece of paper and the pen, a nice ischiomorph, I guess, for an obsolete image that to represent what you're doing. This will launch our model editor. So here I have my model editor. And now one thing you may, if you're actually going to follow along, you may have to do a little bit of resizing. Occasionally there are some screen update issues. If you joggle the size of your bounding box, it'll force a redraw of your screen. Once you're inside of the editor, there is a GUI that will help you build a model. We can go to CC3D project, new CC3D project, and it'll pull up our wizard. We give our simulation a name. Avoid special characters and spaces in the name. If you're doing this for the first time, you need to make sure that your simulation directory is /home/nanoHUB/your home user name. You don't want to be pointing to the root directory. So in my case, I'd select computer J Glazier, choose, and that's a writable directory for me. I just called it new simulation 3. And now I've created a simulation placeholder. I have to say how big the lattice I'm going to do my simulation in is. It's by default 2-dimensional. You make it 3D by changing the Z coordinate. In this case, I'm going to accept all of the default settings for the lattice. So I'll hit next. I then have to say what kinds of cells I'm going to be simulating to give them names. I'll just call them cell 1, add, cell 2, add. I'm not going to use any chemical fields in this simulation, so I'll skip the chemical field specifier. And then I have to say what the basic behaviors that I want my cells to have are, remembering that they don't have anything. I don't tell them explicitly to have it. I'll say they stick to each other, they grow, they divide, they have a volume, and that's all I need for the moment. Now I hit next, and this will generate a template simulation, which I can expand to view by double-clicking here. And here I'm seeing it's a little bit too big, perhaps. I'm seeing the computer simulation code that was generated by the wizard. I can now run this. I can save it by hitting Save All here, or I can do under CC3D Project, Save CC3D Project As work. And if I now want to run it, I can right-click on the name of the simulation and say Open in Player. And if I now switch my tabs back to Player, I will see the executing simulation. I don't know if anybody was able to do that along with me. I know several people have seen this so many times. They could probably do it in their sleep, but probably for a few of you, it's the first time seeing that. Let me just restart the simulation. You see an initial lattice of cells of two types. If I step that simulation, I'll see there's an initial relaxation of that initial condition. I'm going to run it for a bit. You'll see the cells are growing. I can zoom in. You'll see I have my two cell types, cell 1 and cell 2, different colors. If I step it, you'll see that they're growing. Occasionally, I'll see cells divide, and I'll see that when the cells divide, one of the offspring of the division has the same color as the parent, or is the color of the parent, and the other one takes the opposite color. And so there's an alternation of colors as the tissue grows. This is a very simple simulation of essentially an exponentially growing tumor and a little bit of differentiation. Okay. If I wanted to look at actually how the code works, I find that all computational simulations have two pieces. They have a CC3D ML section, which specifies the static properties of the simulation. And then they have some Python code that does dynamics. So things are not fully declaratively specified the way they are with SPML or Antimony. We'd like to pull more and more in to the XML specification as time goes on, and ideas for better ways to do that would be much appreciated. If I look at my XML, I'll see that there's a block of metadata that tells me things like how many processors I use simultaneously, because CompuCell is intrinsically multi-threaded, how big my lattice is. Then I have to specify my cell types. Remember, I had to list the types of cells I was going to be using. Plugins are the basic properties of cells. Here I'm saying cells have a volume. Here's a plugin which specifies the contact energy, the adhesion energy between cells of different types. And then there, at the end of the XML, there's some very simple tools for specifying initial conditions. These are called steppables. They're executed not in micro steps, but once every macro step of the simulation. Uniform initializer draws a square, that square lattice of cells. I can specify how big the lattice is, whether the cells are touching each other or not, how big the cells are, and whether I have one cell type or two cell types and what the ratio of how many cells of each type I have. I can draw more complex things by overlaying multiple simple geometries. If I switch to my Python, I see a bunch of boilerplate. The two key ideas that are important is that inside of these Python steppables, you're going to have a start function that's called once at the beginning of the simulation. You're going to have a step function that is executed every time step in the simulation. And then sometimes you're going to have, there aren't any in this particular case, stop function, end functions or interrupt functions that are called at the end of a simulation to do things like save outputs. In the start function, we have to specify how big our cells are and how compressible they are. So we have for cell and cells.cell list, typical thing to do, iterating over all cells in the simulation. We have something that controls the cell volume. At each time step, all of our cells have the target volume increased by 1. That gives us exponential growth, which isn't biologically realistic. We could very easily change that to mean more meaningful. And then once we've got our cells growing, we have to come up with a rule for when cells are going to divide. In this case, we have a simple volume rule. It says that cells divide when they get to a volume of 50 voxels. When they divide, we have to say how they divide. They could divide on random orientations. They could divide along major axis, minor axis, fixed axis. And then we also have to say what happens after cells have divided, what fraction of the volume is inherited by the two daughter cells, whether the daughter cells have the same attributes as the parent. Here we're cloning the parent into the daughter cells. And then here we're doing that little bit of an update of the cell type where one of the daughter cell has the opposite type to the parent. And that's really about all the complexity that you need in these kinds of simulations. Of course, they can get quite long when you're doing models with lots of cell types, lots of chemical fields. Analysis code can be long, but the actual model specifications aren't that much more complicated than that. Coming back to the things that people are interested in here, Computational allows you to run SBML models, CellML models, Antimony models, and MaBOSS models, either as freestanding models or as, I'm going to go back to the full screen for the PowerPoint, so it's a little bit easier if you can see. Either as individual cells or connected to the whole model as a whole. This is an example where you can actually pull in directly from the biomodels database code. So we specify actually the address of the biomodels database model, pull it in. It's a Tyson cell cycle model in this case. I load that into my individual cells. I run that inside of Computational. So each of these cells has an independent cell cycle model. If we remember Tyson, cell division is triggered by a particular ratio of chemical, particular concentration of cyclins. That concentration depends on the amount of the chemical and the volume of the cell. So as the cell grows, the concentration goes down for a given amount, and that triggers cell division. A little bit more interesting, perhaps, would be delta notch patterning. Here we have a simple, very simplified version of delta notch inside a single cell. Written here as OD, production rate of notch, production rate of notch intracellular domain and delta. Those happen inside of a cell, but what's interesting with delta notch is it's juxtacrine signaling. Delta and notch happen primarily as signaling between adjacent cells. And so here I've specified what's going on within a given cell in Antimony, loaded that into every cell independently in the simulation. So here actually I'm doing the loading here. Self.add Antimony model to cell. That'll load an independent copy of that model into each cell. I can read and write the values of the Antimony model through a Python dictionary, which is automatically attached to each Computational cell. And then I have to couple the values of the delta and notch within a cell to its neighbors. And I use this CompuCell function for a neighbor common surface area in neighbor data list that allows me to identify my contact area with my neighbors. Then I can pull the values of delta and notch from my neighbors and send the signal back and forth. So I don't have an automatic way of wiring up the networks between adjacent cells. It would be very nice if we could find an SPML extension that would allow us to do this using something like an Antimony declaration or an SPML declaration rather than having to do it in Python. And if I put that together, this is what I get. Here I'm looking at the delta level. And I'll see the patterning developing spontaneously in the tissue. So those are some examples of the kind of patterning you can do here. MaBOSS is a Boolean, stochastic Boolean network simulator. It is relatively new inside of CompuCell, but again, you have the same basic structure. I can load a MaBOSS model, in this case, a cell cycle model, and also a delta notch model into each cell. I execute the MaBOSS model inside of each cell. I do the same kind of exchange of information between adjacent cells that I did in the previous model. And here it's controlling delta notch is not only controlling the differentiation state of the cell, but also its cell division. So that gives you a couple of ideas, things you could do. Clearly, compared to doing network models on their own, network models are complicated enough. When you start doing these multiscale models, you have lots of components, lots of component types, lots of parameters. The mapping of the behaviors to measurables isn't always as obvious as you might wish. And so these are not trivial models to build or validate. And to understand what the outputs mean can be a little bit challenging. And of course, they're big models, so they can take a long time to run. Is it worth doing this kind of virtual tissue modeling for your problem? Well, the key thing typically is whether you need spatial information or not. If population level homogenized models work, they run faster and they're a lot simpler. Sticking to Antimony is great if you can get away with it. Secondly, how many cells do you need? If you need to model 10 to the 9th cells, you're probably out of luck and you're back in a continuum world. If you can get away with thousands or hundreds of thousands of cells, and the phenomena you're looking at depend on hundreds of thousands of agents, then it's practical to model each cell individually. If the cells are moving, that usually means that you need to go to spatial models. So if you have immune response, metastasis, vascular growth, these are situations in which network models by themselves typically don't give you all the information you need. You need to have some basic hypotheses about signaling, regulation, transport, behaviors, interactions. These models are especially useful when you expect feedback within and between scales and between agents to be critical. If your agents are behaving independently, you typically don't need this kind of modeling. And then if you need a lot of detail about cell shape, subcellular organization, or ECM, then usually these are problems that are just too hard to solve with these methods. We'd love to work with people. We're always looking for people who want to use these methods, who want to build models. We're definitely looking for people who want to help us extend the tools, help us build new modeling languages, or at least extend modeling languages that make the model specification better. That wizard could use a redo to make it more sophisticated and to make the building of the simulations a little bit easier. If you're interested, we would love to talk to you. I should say that one of the things, if you're interested in specifically in the infectious disease work, We have a NIH working group, which you'd be welcome to join, that's talking about this kind of modeling and other kinds of modeling as well. Herbert's spoken to that group. We've had Paul Macklin speak many times. So if you're interested in that, we would be happy to have you join us. Computational is designed to be modular and extensible. It's all DLLs. And so if you're interested in working with us to make it better, we'd be very grateful. There's a lot to do to make it better, and we'd appreciate your help. Again, if you want to try downloading it, you can get it from computational3d.org. If you want to run it online, you can do it at nanoHUB. I encourage you to think about nanoHUB as a tool for deploying and distributing simulations in a shareable way. Many of you may be coming back, hearing the same material yet again in 2 weeks for the Computational portion of the summer course. You're welcome to join us August 1st to August 7th. If you haven't joined the Computational course and you want to, even though registration is officially closed, we won't turn anybody away. We want to thank our funders, especially the joint grant we have with Herbert. And I need to disclose, since I mentioned that we mentioned a little bit remdesivir, that through my pension plan, somebody bought some Gilead stock. And so I have to disclose that conflict. So I hope that wasn't too much for you. I know you're coming off the end of a long week and you've seen a lot, but I hope that that was interesting for the few of you who haven't seen it before. And I'd be happy to answer some questions.
Hi, Dr. Glazier. Thank you so much for your presentation and for persisting despite having COVID. That's kind of above and beyond.
Yeah.
I was wondering, you might have mentioned it and I might have missed it, but for the XML format you have for Computational 3D, is it specific to Computational 3D or is it broadly being developed by like the multicellular modeling community?
So we have, in a sense, we're prisoners of having been first movers. So our XML is, in fact, Computational XML. And other people like Paul now has an XML that he's developing that's to a large extent based on what we did, but it's gone beyond it. And I think it probably is really time to go back and to try to develop a more generic specification standard for multicell modeling. One of the problems that we face when we want to build declarative languages for model specification for multicell models is that it's harder to separate the agent behaviors and their specification from their implementation methodology. In the case of network models, when you draw A plus B goes to C at rate K times A times B, that doesn't care whether you're using a Gillespie solver, an ODE solver, or something else. However, when I write a simulation of a cell and I have a cell agent, if I have a center model, the cell has a position, but no volume, no surface, no shape. If it's a Computational GGH CPM type cell, it has a volume, an explicit shape, no explicit position, and no explicit surface. If I do the kind of finite element model that the Moffat people do, then I have a lot of additional information. If I do a vertex model, I have a explicit surface, but no volume. And so specifying the behaviors and properties of the cell in a method-independent way becomes a little bit more difficult. In principle, I can abstract and say the cell has a volume, And now it's up to my methodology to interpret that volume in an appropriate way. That's okay for static properties like volume. It's a little bit harder to do when I have interactions. So things like, for example, cell adhesion. There's no fundamental concept of contact energy in a center model. There's center to center attraction, but there's no surface area dependent attraction in center models. And so one of the fundamental concepts that exists in a POTS model simulation like Computational, which is contact energy per unit area, doesn't exist in Herbert's Physicell. On the other hand, concepts like cell cycle, cell differentiation state, these are things that are very universal. The other thing that you get, which is a little bit difficult, is that in our modeling language, you tend to specify things by mechanism. You specify all of adhesion properties together because the physics is done together. But if you want a declarative language where you can build a cell type, you want to have the cell adhesion properties of that cell bound to the cell, not to the universe that they live in. And so you have to cut the model specification up in a different direction. And that can be a little bit tricky. Getting good defaults and things like that can be a little bit tricky. So that's a very long answer, but I think it's a very fundamental question, and I think it's critical to go forward with this. We spent years and years and years talking about it, and we need to do it. I'd love to do it. I'd love to get that done.
We need to convince someone to pay us to do it.
That's always the hard part.
Right.
Naveed, you can ask your question.
Yeah, wonderful talk, James. I was wondering if I mean, you brought up the some kind of like finite element software that is out there. I was wondering that is there any software that particularly uses finite element for continuous models that is compatible with Computational, and this communication is like simplified? Because I have asked this question from you before, and you said someone in the past years has tried to like make, I don't know, Phoenix and Computational talk to each other. But is there like an app that actually makes this easier to have like this kind of models talk to each other?
Well, so one thing about Computational is that it's Python. So I'm sorry, this is the kind of thing where the COVID is getting me. There is a finite element solids modeling Python library whose name is escaping me at the moment. It's not particularly a biological one. It's more for solids modeling. It's a little bit slow, but it's a nice library. And people have published simulations where they'll do an import of that library into Computational, and then use that to solve the properties of extracellular matrix deformation, for example, and couple it to Computational cells. Since Computational cells are all since Computational is all built around the idea of forces and effective energies, you could write a coupling term between a Computational cell and an object living in another methodology, and provided that they send either an energy or a force back and forth, you can have them influence each other. So there are some papers in that list that I showed where people did couple Computational to external finite element libraries that were Python derived ones, not Phoenix.
Okay, thank you.
Well, I don't see any more questions.
Okay.
But I do have a comment. So I've worked in Dr. Sarah's lab for a while, and I've always used Antimony in the context of Epithelial, so I was really like surprised to see it in the context of Computical. I thought that was really cool.
Yeah, well, we love it because it's text-based. And so when you write your CompuCell simulation, your antimony is easy to read. You have it right there in the simulation. Now, importing SBML is great. Importing MaBOSS is great, but then you need to use a separate model editor to be able to see it. And the great thing for me about Antimony is that you can read the simulation specification in line, and you can edit it in line. And so while functionally the integration on the back end doesn't care where it comes from, in terms of model specification, building Antimony models inside CompuCell is really easy. There are a couple of things that you typically don't do when you're running network models by themselves, which we do a lot. It's one of the reasons I talked about Delta Notch, which is we run instances of the same model in multiple cells and then cross-couple them. So we do that a lot. We can load multiple models into an individual cell. We could have an Antimony model of metabolism, an Antimony model of cell cycle, an Antimony model of differentiation, and those can all be loaded independently into a single cell. And again, any Antimony variable or parameter is exposed through the cell dictionary, which is a standard CompuCell concept. And so you don't have to do anything fancy to read and write and control Antimony. Just the way from Tellurium you write r dot variable name equals something. In our case, you'd write r dot SBML. Unfortunately, we use SBML for Antimony as the name. Our cell dot SBML model dot model name dot dict square bracket variable name equals something. So it's a little bit longer to type, but you don't have to do any getting or putting. Everything is natively available. And so we have the same ability to manipulate. That's because libRoadrunner is so good. libRoadrunner weren't so good, you couldn't do all of that.
Well, seeing that there's no more questions, can we all give Dr. Glazier another virtual round of applause?
Okay, well, thank you for having me. I appreciate it very much. I hope
Great.
the rest of your, last few hours of your workshop are really productive, and I hope I'll see a few of you in 2 weeks for the CompuCell workshop. Okay.
Hopefully you're
Thanks
better
so
by
much.
then as well.
Oh, I better be. If I'm not, I'm going to be.
God, terrible.
Okay, thanks, everybody.
Okay,
Bye.
great. Bye now.